Hi, I'm Uday Patel.
And I'm Josh Clark, and today we'll be talking to you about our project effects of herbicide glyphosate on DNA methylation in human osteoblast cells.
Our general outline for this presentation is background, research question and hypothesis, methods, and results.
So what is glyphosate? Glyphosate is the main component of the herbicide found in Roundup. It is the most commonly used herbicide today. 1.8 million tons of glyphosate was applied between 1974 and 2014. It is an inorganic phosphorus compound that's found in our non-organic fruits, vegetables, and soy products. It inhibits a plant enzyme involved in AA synthesis. So where is glyphosate found in our environment? Glyphosate is actually found in 85% of the foods that were sampled. High levels of glyphosate is found in honey. They also detected in 36% of 154 water samples collected in the Midwest in the United States. They found both glyphosate found in both ground and surface water. It was detected in soil, crop products, domestic and wild animals. Exposure to glyphosate is widespread. So what are the health effects of glyphosate? Glyphosate is known to alter kidney and liver function. It disrupts the maturation in rats and fish and also increased developmental abnormalities. It is a possible carcinogen. It also may cause DNA damage and increase oxidative stress, which may lead to epigenetic changes in DNA methylation and altered gene expression.
What are epigenetic modifications? Means above the genome, doesn't involve changes in DNA sequence, can include changes in DNA methylation and histone modifications, known to lead changes in gene expression, also can contribute to cancer progression and alter cell function. So DNA methylation and gene expression. Methylation occurs on the C of a 5-CpG, which is right here. Little 5-MC found in gene promoters. Increased DNA methylation in promoters can reduce gene expression. Our research question is, does chronic exposure to glyphosate alter DNA methylation in human osteoblast cells? The hypothesis is the level of DNA methylation is altered in human osteoblasts chronically exposed to glyphosate. The significance is the function and activity of normal osteoblasts and bone dynamics in humans exposed to glyphosate may be altered due to epigenetic changes.
Our modeling system that we use for this experiment utilizes the hFOB 119 normal human osteoblast cells. Osteoblasts are responsible for bone formation and remodeling. Our cells was isolated from the femoral bone tissue of either the hip or the knee. During development, osteoblast goes through a process called osteogenesis, which form osteocytes. Our cells were exposed to different concentration levels of glyphosate over a 2, 3, and 4 week period. Cells were collected at the indicated weeks, and PBS was used to wash the cells, which were then scraped and pelleted. The pellets were then frozen at -degrees Celsius.
Our second part was DNA isolation. DNA was isolated from frozen cell pellets using PureLink Genomic DNA kit. DNA concentration was then determined using a qubit nanodrop reader. Purity was indicated by a 260-280 ratio. Now we did the 5-MC assay. We used the Zymo Research 5-MC DNA Elisa kit. Each DNA was analyzed and repeated. Standard curve was generated. OD values were standardized again. Averages, standard error, and significance was determined.
The y-axis of our results represents the OD reading per nanogram of DNA of the sample. The x-axis represents the weeks the cells were isolated, and the asterisk indicates the significance. It can be seen, except for one sample at week 2, that there is no significance between the treated and untreated for both weeks 2 and week 3. However, at week 4, we see that the DNA methylation significantly increases for all 3 treatment groups, indicating that there is indeed a dose-dependent increase.
So these results show that culturing the cells for an extended cause increases in DNA methylation independent of treatment, which is shown in this figure below right here.
In our conclusion, Human osteoblasts exposure to low levels of glyphosate for at least 4 weeks have increased levels of DNA methylation. At week 4, the increase in DNA methylation is dose dependent, which with the highest concentration of glyphosate showing the greatest increase. Extended cell culture of hFOB 119 osteoblasts displays an increase in DNA methylation independent of glyphosate exposure.
So our future plans is determine the genomic locations of the changes in DNA methylation, also determine if and what genes are located within regions of altered DNA methylation. This is the end of our presentation, and Josh and I would like to thank UNCG Department of Biology for our undergraduate research funds.
Thank you so much.
Thank you.